Hi, my name is Sandra, and today I'm going to be giving you a brief introduction as to what the MMR signaling pathway is. The MMR signaling pathway, also known as DNA mismatch repair, it plays a crucial role when it comes to ensuring the genomic stability in our body, and it also plays other couple of crucial functions as well. As stated, it recognizes pairs that are a mismatch in our DNA sequence, and this could be due to errors that happen during the insertion and deletion process, especially during DNA replication. Not only that, but it also triggers apoptosis when the DNA damage in a DNA sequence is beyond repair. Furthermore, it's also likely that the MMR signaling pathway is a linkage to cancer suppression, but this can be more applied towards colorectal cancer. Lastly, the MMR signaling pathway, its proteins also help contribute to antibody diversity. And this is because it processes different DNA mismatches, especially during the differentiation of B cells in our body. Now, towards my left is a general flowchart as to how the MMR signaling pathway is supposed to look like. And I know it looks a little bit confusing, so therefore I'm going to be passing it over to Nivita to give you a further breakdown as to the components of the MMR signaling pathway.
Hi, my name is Nivita and I'm going to bring you guys through the key steps. So the first one is actually mismatch recognition, which is initiated by the MUT-as homolog proteins. In eukaryotes, the MSH2-MSH6 complex or the MUT-as alpha complex recognizes base-base mismatches and smaller insertion-deletion loops, while the MSH2-MSH3 complex or the MUT-as beta complex recognizes larger insertion-deletion loops. The next step is actually recruitment of MUT-L homologs. So after the initial mismatch detection, MUT-L homologs are recruited. In eukaryotes, MLH1 PMS2 MUT-L alpha plays a critical role, coordinating with downstream processes like DNA excision and repair. The MUT-L proteins help orchestrate the rest of the repair process by facilitating interactions with other proteins. And then we move on to excision of the error. So after the mismatch recognition, the mismatched DNA strand is excised. In prokaryotes, MUT-H is an endonuclease that cleaves the DNA strand near the mismatch. In eukaryotes, exo-1 or exonuclease 1 is responsible for excising the mismatched strand, although exo-1 independent pathways can also function when necessary. And then we move on to DNA resynthesis. So after the excised DNA is removed, DNA polymerase fills the gap with the correct nucleotides using the undamaged strand as a template. In eukaryotes, DNA polymerase gamma is typically responsible for this step. So the final step is ligation, where DNA ligase 1 seals the remaining nick in the newly synthesized DNA strand, completing the repair process. Finally, we go on to proofreading and crosstalk with other pathways. This is when the MMR system also interacts with other pathways like base accession repair or BER and participates in cellular processes like apoptosis if the mismatch is not repaired. MSH2 in particular has roles in regulating cell responses to damage and promoting apoptosis when repair is inadequate.
So now I'll be explaining about the different types of consequences that will be faced when there's a defect in the MMR signaling pathway. The first consequence would be there will be an increased mutation and cancer development. So a malfunctioning MMR pathway is commonly associated to microsatellite instability. And if you're unaware as to what microsatellites are, they are short and repetitive DNA sequences that are found in the genome of organisms. Now, this condition basically refers to the accumulation of the errors in these sequences, and this is often observed in various types of cancers, specifically in colorectal cancer. The second type of consequence would be cellular instability. So, as I mentioned earlier, the MMR signaling pathway also helps trigger apoptosis towards cells that have DNA damage that is beyond repair. Now, a malfunctioning MMR system may actually allow these damaged cells to escape apoptosis, and this can actually lead to the accumulation of genetically unstable cells in the body. Lastly, your body would also generate a resistance towards chemotherapy. Now, chemotherapy relies on using agents such as temozolomide and platinum-based drugs in order to induce DNA damage. Now, cells that are deficient in MMR actually fail to recognize and to respond to the damage that are supposed to be caused by these agents, and usually allowing them to survive chemotherapy treatments which would otherwise kill like MMR proficient cells.
So these are a few solutions that can be suggested. So the first one is actually gene therapy and editing, where gene editing techniques like CRISPR or Cas9 are being researched to correct mutations in MMR genes, such as MLH1 or MSH2. These technologies could potentially restore proper MMR function, though clinical applications are still in early stages. Next, we have immunotherapy, where immune checkpoint inhibitors or ICIs, such as pembrolizumab, have shown remarkable success in treating cancers with MMR deficiencies. These tumors tend to have high microsatellite instability or MSI and generate numerous mutations, which can make them more recognizable to the immune system. Studies have also demonstrated that patients with MMR deficient cancers respond well to ICIs, and the FDA has approved pembrolizumab for MMR deficient tumors regardless of their tissue origin. Next, we have targeted therapies where PIRP inhibitors and other drugs targeting alternative DNA repair mechanisms are being tested. By blocking compensatory pathways in MMR deficient tumors, these therapies induce synthetic lethality, leading to cancer cell death. In addition, kinase inhibitors, example targeting ATR or ATM, are being explored to further sensitize MMR deficient cancers to DNA damage. We also have restoring MMR function, where epigenetic drugs that reverse gene silencing, such as DNA methylation inhibitors, could potentially restore the expression of silenced MMR genes, particularly in cases like Lynch syndrome, where epigenetic changes often inactivate MMR proteins. Finally, we have early detection and prevention. Genetic screening for Lynch syndrome, a condition linked to defective MMR, allows for preventive measures like increased cancer surveillance and early interventions. MSI testing and MMR protein immunohistochemistry are valuable tools for identifying MMR deficient tumors, allowing for personalized treatment strategies. So these are the references used to source information for this presentation. And with that being said, thank you very much.